this lecture is all about fertilization, cleavage, and gastrulation. Those are the three major topics, and then we'll move on from there, obviously. These are the initial stages of development. So there are actually 4 things that a species has to overcome in order for members of the same species to even form a zygote. 4 major hurdles that they have to overcome. In some situations, like in the ocean, there are, how do I put this without sounding crass or crude, massive orgies of sperm being released into the environment of, probably didn't do it right, but anyway, there are lots and lots of sperm from a variety of species that's just all over the place, okay? So the question then becomes, how does each species sperm recognize each species egg? Well, that's the first hurdle, species-specific sperm egg recognition. What does that come down to? It comes down to the proteins that are on the sperm and on the protein. If they don't match, then they will not join up with one another. They may touch one another, but they will not go through the next stages of fertilization. So that's one thing that prevents different species sperm fertilizing different species eggs, is because of these proteins that are found on the surface of both the sperm and the egg that are involved in species-specific sperm egg recognition. Once the sperm and the egg have found one another and have the right mojo, or proteins, then you get regulation of sperm entry into the egg. Now, for humans, as well as many other species, there typically is an acrosomal cap which has a variety of digestive enzymes that are necessary to penetrate the zone around the egg. Okay? There is a number of things that happen, though, to regulate sperm entry. One of the biggest problems that occurs is polyspermy, which is multiple sperm fertilizing one egg. You've got to have just those 2 haploid cells fusing to become a diploid cell. Any more chromosomes than that, you have massive problems. Now, in the case of plants, they can tend to, they overcome polyspermy much better than vertebrates and invertebrates. How do you prevent polyspermy then? There's actually 2 major events. There's a fast reaction to prevent polyspermy, and then there's a slow reaction to prevent polyspermy. Once the digestive enzymes from the acrosomal cap start breaking through the zona pellucida, which is just a huge barrier outside of the oocyte, then once the sperm enters in, once one sperm enters in, the first thing that happens is there's a rapid repolarization of the membrane. They typically have a membrane polarity of -70 millivolts. As soon as the sperm enters in, then it actually changes the ionic composition around it, and it reverses the polarity of the membrane, so that's about 20 millivolts. This creates a negative, or kind of a repulsion of the sperm. Now, this is what's called the fast reaction. But this doesn't really, in fact, you can still get polyspermy. And so this is more of one of those, hey, I'm going to hold off all the sperm until I can get to the slow reaction. And here's the slow reaction, the cortical reaction. The cortical reaction is what seals the deal to prevent polyspermy. What happens is there are these granules with these enzymes that when you get this depolarization, initiates these granules to release their enzymes, and what it does is starts chewing away parts of the membrane so that the zona pellucida and the membrane of the oocyte just become immensely separated from one another. So basically, there are enzymes that are cutting what's holding the zona pellucida and the oocyte together, and it's then filling with fluid and just massively separating so that all the other sperm are out here, and this just pulls away from that and prevents further sperm from entering in. So that's the cortical reaction. You get these calcium waves that go through and initiate this enzymatic cutting away of the membrane from the zona pellucida, and that's the second thing, is Once the sperm penetrates that, you got to prevent other sperm from entering in, and it goes through that fast and then slow process of prevention of polyspermy. Then you've got to fuse the nuclei. We usually think that that's kind of a simple thing, but it's not quite the easiest thing in the world to get those nuclei to fuse together. One of the interesting things about humans is women's oocytes are still undergoing the second stage of meiosis even at the moment of fertilization. And it will not finish meiosis 2 until fertilization occurs. Then it will get rid of the last pronucleus, or the last polar body, and then you'll get fusion of the two nuclei of the sperm and the egg. So even after a woman has ovulated the oocyte, she actually still has two nuclei, two haploid nuclei in the cell that are finishing up metaphase, and they actually kind of stop at metaphase. Sperm fertilizes the egg. It finishes metaphase 2 and anaphase 2, and then telophase 2 gets rid of one of the polar bodies, one of the nuclei, one of the haploid nuclei, and then you get fusion of the two gametes. And then the last thing, and this is kind of in conjunction with the sperm entry, the cortical reaction doesn't only prevent polyspermy, but it creates these calcium waves that can actually initiate protein synthesis. Remember that the oocyte has these mRNAs that are sequestered to various areas in the cytoplasm, and you need some type of trigger to tell the cell when to turn on translation of these already transcribed mRNAs. Well, that's part of it as well. So the actual fertilization event initiates the translation of these, and sometimes it binds to proteins that have acted as repressors, removing them from that, and then all of a sudden they can be translated into, these mRNAs can be translated into proteins. And then that will then get the ball rolling. It'll start causing gene transcription and translation to occur in the new nucleus and the fused nuclei between the sperm and the egg. And you get the subsequent events that occur from that. Let's look a little bit about the Xenopus. One of the interesting things about frog embryos, and in fact that's what's illustrated here in this, is the fertilization event isn't just about fusing the nuclei, because in some cases it actually matters where the sperm enters and what happens subsequently. For example, in the frog embryo, normally it's like this. The top half of it is actually dark, just like this. It's not pseudocolored. This is what a frog embryo looks like. You have a dark area on the top here. You have a lighter area down beneath here. Well, the sperm can only enter into this top part. The reason for that is because this is what's called the animal pole. It has a lower concentration of yolk. And so the way in which this is configured, sperm can't come in this bottom part at all. It can only come in this top half part right there. Now, as the sperm enters in, that actually predetermines the axes of the Xenopus. In fact, after the sperm enters in, you get what's called a cortical rotation, where this will then move about a 30-degree angle. Now, later on, we'll talk about the relevance of this, but I'll give you a prelude to it. In this top half, initially, there's an even distribution of genes and mRNA, such as disheveled. But when this rotates, it actually rotates the concentration of this, and you start getting disproportionate areas. You'll start to see how cleavage will actually generate different amounts of protein in these group of cells and in these group of cells, and that's vital to the overall development of the Xenopus. Like I said, things are not distributed evenly in the initial oocyte. So this leads us to the second concept, which is immediately after fertilization begins,